Hello, everyone. Welcome to this Symposium, a tale of parcels and gradients, individual differences, and behavior associations. So we propose this symposium in order to discuss two different views on brain organization, parcellations and gradients. So in this symposium, we seek to reconcile these two views with a specific focus on modeling inter-individual differences in brain organization and cognition. So resonance functional MRI is one of the key tools to capture brain organization in single individuals. So it lends a powerful window to intrinsic functional brain organization. So since the early days, resonance functional MRI analysis have focused on deriving brain parcellations. But recently, several analytical methods have emerged and exploit more continuous representations of brain, which is known as gradients. We know that individual differences exist in brain organization. Using parcellation or gradient techniques, we are able to obtain individual-specific representations of our brain. Based on these individual-specific brain representations, we can then explore whether they can provide insights into the relationship between brain organization and cognition. Today, we will discuss improved methods to categorize human organization, and we will also discuss whether these brain representations can better capture inter-individual variability, and ultimately provide new insights into the brain basis of cognitive functions and brain disorders. I think it should be fun today to walk through that together. We're going to have 4 speakers today. We'll start by Tian from the University of Melbourne, talking about different techniques for representing brain organization. So smooth gradients or discrete regions or a bit of both. Followed by me talking about comparison between gradients and parcellations for resonance functional connectivity behavior prediction. Jonathan Smallwood from the University of York talking about macro scale brain organization and cognitive dynamics. We will finish with Oualid Benkarim from McGill University talking about studying idiosyncratic connecting organization using gradients and parcellation techniques in typical and atypical development. So let's welcome our first speaker, Tian.
Hi, everyone. I'm Tian from the University of Melbourne in Australia. I have been very excited to be part of this symposium to tell the story of brain parcels and gradients. So I think I'm the first speaker in this symposium. So as the first storyteller, I will introduce the two representations of brain topography, smooth gradient, discrete regions, or a bit of both. So when we talk about brain topography, we mean that the structure, function, and connectivity of the brain, they are topographically organized, in contrast to diffuse or random phenomena. So classic models of brain topography parcellate the brain into distinct structure and functional regions, where recent cortical gradient studies suggest that the brain and the topographic variation of the brain might be better conceptualized in terms of a set of continuous gradients that overlap in space in the absence of discrete boundaries. So this new idea challenged the traditional notion of brain topography and gave rise to some contention about whether the brain is organized into discrete partitions or continuous gradients. So here are the two models of brain topography. Are the two models really contradictory, or can we somehow reconcile the two models? So yes, sure, we can continuously characterize the spatial variation in brain structure, function, or connectivity across the topography, but we should also be able to delineate a hard boundary when the spatial variation is significantly large. So this led to the question of mode selection. How sharp a change must be to warrant such a hard boundary. So in today's few minutes talk, I will briefly introduce principled approaches to reconcile the two topographic representation of the brain. In particular, I will show how these approaches can be or have been utilized to characterize the topographic organization of one of the brain's remaining unknown territory, the subcortex. So while we have particularly focused on the subcortex, the reason is actually quite simple, because as you can see in previous slide, the cortex has been most extensively studied whereas the subcortical structures are widely implicated in many neuropsychiatric disorders. But many of their subcortical regions have been studied in isolation without first establishing functional boundary between them. The very few existing connectivity-based atlases, including more than one subcortical regions, generally do not preserve the established anatomical boundary between them. So, okay, we think maybe this suggests structural function divergence, or maybe need some new methodologies to map the topography. So here in our study, instead we expect to see a rudimentary alignment between the anatomical and the functional architecture in the subcortex. But we would also expect to see functional subsystems that potentially divergent from the anatomy. So what we did, we first used established methods to map the principal gradients in functional connectivity across the topography of the subcortex. To better characterize the connectivity gradient, we developed a technique called gradientography. Let's see how it works. So here we have the principal gradient map or eigenmap. We can then estimate the gradient orientation for each subcortical region. And then we feed tensor to each of the subcortical voxels in the estimated gradient field, such that the shape along the long axis of each tensor indicate gradient direction, whereas the gradient magnitude is represented by the length of the tensor. So why we feed the tensor? Because here, look, millions of streamlines can be propagated in the tensor field, in the same way as white matter tractography is typically performed in diffusion imaging. But here we proposed tractography that is based on the connectivity gradient, so we call it gradientography. Let's take a look at all those streamlines. Each streamline represents a trajectory or maximum change in the eigenmap. Here we color the streamlines according to the gradient magnitude value of each subcortical voxels. So when this kind of locations with circumscribed band indicate local maximal inner gradient magnitude, which suggested the potential functional boundary there. So the technique of gradientography not only enable a striking visualization of the brain connectivity gradient, but also provide identification of the locations of gradient magnitude peaks. So let's an even closer look. Here are the streamlines traveling through the basal ganglia. We can see transitions between the globus pallidus, the putamen, nucleus accumbens, and the chordate. We can then parameterize such transitions using the diversity curves. So you can see the 3 local peaks in the diversity curves corresponding to the locations of the functional boundary between those subcortical nuclei. So we can see a similar phenomenon in the ventral part of the subcortex, where streamlines traveling past the left-right thalamus, hippocampus, and amygdala. So here, more local peaks are noticed in the diversity curve. Now we are asking, shall we consider all the peaks? Would that cause overpopulation? To what extent the peaks can be explained just by chance, or the other confounds, such as the spatial smoothing during data pre-processing, or the construction or geometric contraction. So to control those factors, we generated such a null model as the curve shown in gray. And we parcellate the subcortex at locations only when the empirical peaks are significantly larger than the peaks in the null curve. So if the null can be rejected, we parcellate the subcourse at this location. Otherwise, we argue that the topographic variation in functional connectivity is best represented as a continuum in the absence of discrete boundary. So this model selection process yielded an initial parcellation for the subcortex, comprised of 8 bilateral regions, so 16 in total, which corresponded very well with the no anatomical nuclei, as you can see in this slide. So then we map the connectivity gradient for each of the 16 regions separately, with the performing of model selection recursively, yielding a hierarchical parcellation for the subcortex. However, look, there are 4 scales of organization, but not all subcortical nuclei comprise 4 scales of this organization. Let's take, for example, we have the globus pallidus, This globus pallidus only extend to scale 2, whereas hippocampus and thalamus, they extend further, comprising 4 scales. So this is actually very consistent with the greater functional and organizational complexity within the thalamus and the hippocampus compared to the more specific role of the globus pallidus in the regulation of movement control. So in a brief summary, we proposed a model selection framework to reconcile the two representations of brain topography to disambiguate smooth connectivity gradients from discrete area boundaries. So we show a hybrid representation of the subcortical topography. And this multi-scale subcortical atlas can be found on GitHub. It's free you can download them. And also check this preprint if you are interested to see how subcortical topography reconfigures in response to changing cognitive demands and how it relates to the intrinsic organization of cortical networks and how subcortical functional connectivity explain behavior variance. So finally, in this last slide, I want to briefly show an example brain regions where the two topographic models are competing to each other. As you can see on the left side, the number of insular subregions varies substantially across previous population studies. However, we found that the topographic variation in the insula is best modeled as a continuum along an anterior-posterior axis with weak evidence indicating a sharp boundary in this continuum. This continuum is also consistent across several imaging modalities and early work of track tracing in macaque, those non-human primates. So, all right, to close today's talk, I really want to say that try model selection and let the statistics tell you how to best represent the brain topography. And thanks everyone for listening, and thanks all the collaborators. And any questions?
Hello everyone. In today's talk, I will compare gradients and parcellation approaches for resting state functional connectivity behavior prediction. So the brain is active even at rest. This video from Daniel shows the functional MRI signal when the subject is lying at rest in the scanner. As you can see, these large spontaneous activity fluctuations appear throughout the brain. So these spontaneous fluctuations during resting state functional MRI can be used to interrogate brain network organization. For example, if we choose a seed in PCC and correlate its time course across the cortex, we can get this famous default mode network. This procedure is called functional connectivity. So if we move the seed region around brain, we see that the correlation pattern changes a lot, suggesting that adjacent brain regions can have very different connectivity patterns. So this has motivated many studies to extract discrete or continuous representations of brain using functional connectivity. Here, I'm showing you examples of gradients and parcellations. So gradient techniques look for continuous representations of brain, and parcellation approaches can be further categorized as soft parcellations and hard parcellations. For soft parcellation, each vertex can be assigned into multiple RIs, while for hard parcellation, each vertex can be assigned to one single RI. So in these three examples, we can see they all capture similar default mode network structure. Many papers have demonstrated that individual differences in resting state functional connectivity can be used for behavior prediction. Therefore, behavior prediction can be used to assess quality of different brain representations. So in this talk, we focus on behavior prediction and compare the performance of gradient techniques versus parcellation approaches. We consider two gradient techniques, two soft parcellation approaches, and two hard parcellation approaches. I will first introduce two gradient techniques. We first consider local gradient approach pioneered by Steve Peterson's group. The local gradient approach detects the local changes in functional connectivity patterns. So this figure is an example of the gradient map. Bright color indicates rapid change in functional connectivity at that location, and dark color indicates slow change in functional connectivity at that location. We also consider the principal gradients developed by Daniel Margulis, where he applies nonlinear diffusion embedding to decompose functional connectivity into multiple principal gradients. So we align gradients across subjects and pick the top 5 gradients that will match the well across all subjects. For soft parcellation approaches, we include spatial ICA. I guess we all know what is spatial ICA. Basically, spatial ICA decomposes resting state functional MRI data into a set of spatially independent maps and their corresponding time courses. So we try different number of ICA components, 50, 100, 200, and 300. So this figure shows 10 ICA spatial maps as examples. Another soft parcellation approach we include is Profumo. This is a relatively new approach also from the FSL group. Profumo explicitly models between subject variability to estimate spatial maps. So the right figure shows 10 Profumo spatial maps as examples. For hard parcellation approaches, we include the Schaefer 400 region group level parcellation shown here. So the Schaefer group level parcellation is generated based on a gradient weighted Markov random field model, which interrogates both local gradient and global similarity approaches. We also include my 400 region individual specific parcellation shown on the right. So we extended our previous multi-session hierarchical Bayesian model for estimating individual specific functional networks to estimate finer area level parcellations. So far, I have briefly introduced gradient and parcellation approaches we considered for the comparison. As I mentioned in the previous slides, that we focus on comparison between gradients and parcellations for functional connectivity-based behavior prediction. I will then describe our behavior prediction model. So we considered 58 behavior measures from the Human Connectome project, and we use kernel regression to predict behavior. So roughly speaking, what this does is that the behavior of a test subject is estimated to be the weighted average of all training subjects' behaviors. A training subject is weighted more if the training subject gradients or parcellation features are more similar to the test subject. So this similarity is defined based on correlation of gradients or parcellation features between subjects. In this way, we will construct the inter-subject similarity matrix for different approaches. So let's first look at the gradient techniques. For the local gradient approach, the inter-subject similarity matrix is simply defined as the correlation between subjects' spatial maps, subjects' gradient maps. For example, the highlighted entry is the correlation between gradient maps of subject 2 and subject 3. So similarly, the inter-subject similarity matrix of principal gradients is defined as the correlation between the subject's principal gradients. As we consider 5 principal gradients, we end up with 5 inter-subject similarity matrices. So we will use multi-kernel regression for behavior prediction if there are multiple inter-subject similarity matrices. Next, we will talk about the inter-subject similarity matrix for soft parcellations. For spatial ICA, assume there are k ICA components, and for each subject, we can obtain a k by k partial correlation functional connectivity matrix. To construct inter-subject similarity matrix, we took the lower triangular part of the functional connectivity matrix and correlate between each pair of subjects. So similarly, we can obtain an inter-subject similarity matrix for Profumo using partial correlation functional connectivity. However, in the Profumo framework, the inter-subject variability was most captured by spatial maps instead of functional connectivity, as shown in Jennings and Sam's paper. Therefore, we correlate spatial maps for each pair of the subjects and end up with 50 inter-subject similarity matrices. We then average them to obtain a single similarity matrix. So in this way, we have 2 similarity matrices for Profumo, and we then use multi-kernel regression for behavior prediction. So let's move to the hard parcellations. For both Schaefer 400 region group level parcellation and my 400 region individual specific parcellations, we first generate partial correlation functional connectivity for each subject and correlate the lower triangular part of the functional connectivity between each pair of subjects to obtain the inter-subject similarity matrix. So in the last part of my talk, I will show you the behavior prediction results of gradient and parcellation approaches. Here is the behavior prediction accuracies average across all approaches for 58 behavior measures. So the box plot was colored based on how many approaches can predict this behavior very well. And the white box indicates all approaches predict poorly for this behavior. And dark red box indicates all approaches predict very well for this behavior. And the behavior measures are categorized into task performance measures and self-reported measures. We can see there are more orange dots on the left. So if we average self-reported measures and task performance measures, we can see that the task performance behavior measures can be predicted better than the self-reported behavior measures. And if we average prediction accuracy across all self-reported measures for each approach, the pink results are showing prediction accuracies of gradient techniques. The blue results are showing prediction accuracies of spatial ICA with different resolutions and the proformal. The yellow results are showing hard parcellations. We can see that overall the prediction accuracies of gradient techniques are lower than the parcellation approaches, and the individual specific hard parcellation performs the best. We then averaged prediction accuracies across task performance measures. Again, we first see prediction accuracies of gradient techniques and prediction results of the soft parcellations. Finally, results of the hard parcellations. Again, prediction accuracies of gradient techniques are lower than parcellation approaches. So to summarize in this talk, I've shown that individual specific gradients can also be used for behavior prediction. In the kernel ridge regression framework, overall functional connectivity from brain parcellations outperformed gradients for behavior prediction. We have also repeated this with linear ridge regression, and the conclusions are similar. However, we caution that gradient techniques are relatively new, so future research might get further improvements. It is also worth noting that the individual specific hard parcellation approach actually incorporated gradients in deriving individual specific parcellations. Further testing in new datasets and other supervised learners would also be desirable. All our software are available on GitHub. So this includes the Schaefer group level parcellation, our kernel ridge regression model for behavior prediction, and the code of the individual specific hard parcellations will also be released here once the paper is published. I would like to thank our group members and collaborators and our funding support.
Hello, my name is Jonathan Smallwood, and today I'm going to talk to you about my work looking at the relationship between large-scale cortical gradients as a state space for understanding cognition. Before I start my talk, I would like to thank all the people who've contributed to the work I'm going to talk about. These include the members of the Memory and Thought Lab at the University of York that I manage with Professor Beth Jeffries, as well as a number of important external collaborators. My scientific work concerns understanding the neural basis of ongoing thought patterns. These are contextually variable, idiosyncratic across people, and tempodynamic. In other words, ongoing thought varies across places, people, and time. To capture this rich aspect of human cognition, we use the experience sampling method, which allows ongoing thought to be described in different situations. In particular, we can use smartphones to require samples of people's thinking as they go through their daily environment. We can ask people to come into the lab to perform different types of tasks. We can also measure neural activity using techniques like fMRI whilst people do that. Because ongoing thought is a specific state of mind, it requires a particular type of method to analyze the data that's produced. The methods of cognitive neuroscience which we're used to are optimized for understanding the processes in modular regions. However, complex states, on the other hand, are likely to depend on multiple processes. This requires a state space in which different states can be compared. In my talk today, I'm going to focus on the idea that large-scale cortical gradients, or dimensions of connectivity calculated at rest, can act as a state space from which we can understand different types of cognition. On this slide, I'm showing you work that was performed in collaboration with Daniel Margulies and various other individuals, in which we mapped the different patterns of dimensions of connectivity from the Human Connectome project. The first of these, gradient 1, describes the difference between transmodal regions and regions concerned with unimodal functions. The second gradient, gradient 2, describes the relationship between motor and visual processing. The third gradient describes the similarities and differences between regions which we might imagine come on during tasks, often referred to as task positive, and regions that seem to deactivate, including the default mode network. The data upon which my talk is going to be based comes from the results of an ERC project awarded to me as a consolidator entitled Wandering Minds. In this project, we measured people's experiences across a range of different circumstances and tried to make sense of the underlying components using fMRI. One of the approaches that we use is the application of principal components analysis to experience sampling data. We think that this approach allows us to reveal patterns of thought. On this slide, I'm showing you how we can represent one of these dimensions of experience in the form of a word cloud in which the font size describes the importance and the color is the polarity of the item. I'm also showing you word clouds calculated independently from 3 different situations: smartphone data from the real world, experience sampling during laboratory tasks, and experience sampling from those same laboratory tasks in the scanner. And you can see that striking similarities between the word clouds. In each case, the word task is in blue, and words like self and future are in red. This indicates a dimension that goes from a focus on an external task to a type of cognition which we refer to as episodic and social in nature. To understand how these dimensions of experience relate to the brain, we used fMRI to calculate regions that show heightened activity when individuals engage in either types of cognition. On this slide, I'm showing you a region in red in the medial prefrontal cortex, which showed higher activity when individuals engage in episodic social condition, as well as a region of parietal cortex in blue, which showed greater activity when individuals were engaged in external task-focused thoughts. To understand the relationship between these regions, we use functional connectivity analysis, seeding using the 2 regions and identifying the associated networks. You can see that the seeding from the medial prefrontal cortex was associated with many different regions, all of which are parts of the default mode network. In contrast, seeding from parietal cortex revealed a network of regions identified in blue, and which generally correspond to the task-positive systems identified by cognitive neuroscience. This analysis shows that a psychological dimension identified through experience sampling and the application of principal components analysis was also associated with a neural dimension of connectivity reflecting the dissociation between the default mode network and the multiple demand or frontoparietal systems. Since states occur dynamically over time, we wanted to understand how these different neural motifs can emerge. To do this, we used a paradigm in which participants were asked to systematically respond to events in a task, followed by increasingly long intervals where there was nothing to do. Using these periods between the responses as a regressor, we calculated the regions that changed their neural activity with the passage of time. I'm showing you this map on the bottom slide. You can see that some regions increased activity with the passage of time and other ones decreased it. We understood these regions by examining their whole brain patterns and relating these to the gradients calculated by Daniel Margulies and others in his paper in 2016. On this slide, I'm showing you an unthresholded map describing the activity patterns that were associated with increase and decreasing passage of time. These are strongly correlated with the gradient 3 identified by Daniel, which reflects the difference between task-positive and task-negative systems. I'm also showing you the patterns identified using experience sampling that hypothetically refer to the on-task and episodic social state. It's remarkable that both of these sets of maps show similarities, both in terms of the spatial distribution as well as their similarity to the third gradient. This similarity can be seen when we plot these different spatial maps in the 3-dimensional gradient space created by the first 3 gradients. You can see in this map that the state of on task is very close in this space to maps describing decreasing patterns of regions that decrease their activity over time. In contrast, the off-task state is closely linked to regions of activity that increase activity with the passage of time. In conclusion, our work has shown that different patterns of ongoing thought have unique neural motifs. These can be understood as locations in a multidimensional state space that can be constructed out of large-scale gradients. Important features of ongoing experience are represented by the apparent task-positive and task-negative dimension, and these results suggest that we may need to go beyond the view that task-positive systems must be engaged in tasks. Before concluding my talk, I would like to explain that I'm moving my lab from the Department of Psychology in the University of York in England to take up a position at the Department of Psychology, Queen's University in Kingston, Ontario, Canada. If you're interested in working on similar questions than the ones I talk about in my talk, please get in touch either on email or Twitter. Thank you for your attention.
So thank you everyone for prospectively attending to my presentation, where I'll be talking about macroscale gradient analysis for the study of idiosyncrasy in autism. So for the outline, first I will briefly introduce autism and macroscale gradients, to then move on to present our framework and the results, and to finish with some concluding remarks. So autism is a disorder that encompasses a wide range of neurodevelopmental conditions such as language deficits, reduced social interaction, and repetitive and stereotypic behaviors. From the neuroimaging perspective, we can find several studies that report atypical connectivity patterns in autism. However, many of them have yielded inconsistent results due to the hyperconnectivity and hyperconnectivity presented. But this is mainly due to the fact that in autism, there is a high idiosyncrasy, meaning that there is a huge variability in the connectivity patterns in autism as compared to the typically developing. So in our work, we go into use macroscale gradient analysis. Here, gradients are a good approach to describe the brain's organizational principles in a single continuous manifold, instead of using hard or crisp parcellations of the brain. So our purpose is to understand the connectivity alterations that exist in autism, identify the idiosyncrasy differences between the typically developing and autism, and provide accurate and reliable descriptors of these idiosyncrasy. So our framework is composed of the following steps. First, we build the connectivity matrices for each individual, and also for our reference matrix, which we use to compare all the individuals to. And then we use these connectivity matrices to create our gradients and build the embeddings, which then are aligned to this reference embedding to be all in the same coordinate systems. Once we have all the gradients in the same coordinate system, what we do is we cluster to get the so-called EO networks, or the intrinsic connectivity networks, that we're gonna later on analyze, as we can see in the results. So for the results here, what I'm showing you first are the probability maps of each of these intrinsic connectivity networks, as you can see in the left for typical developing and the right for autism. And the first thing we notice is that there is more spread in these probability maps in autism, as you can see, for example, here in the default mode and here in Smallwood network. So if we take a look at this from the entropy point of view, we can see that in these regions, actually, the entropy is higher in ASD as compared to the typical developing here, for example, in the Smallwood network, and here in the default mode, for example. Taking a look at the distributions of these vertex-wise entropies, we can see a clear shift to the right in the distributions, meaning there is higher entropy in autism. And you can see, for example, in these four networks, in the dorsal and ventral attention network, the default mode, and Smallwood network, where the shift is clearly seen. If we take a look at the cortex, at the whole cortex, we can see that the density of this distribution in autism is mainly concentrated to the right, showing that indeed there is a higher entropy and therefore idiosyncrasy. So, as I mentioned before, this spreading of the probability maps is due to the fact that there is a huge variability in the spatial locations of the networks, and this is due to the spatial shifting of these networks. So, in this part, we're going to present two descriptors of this idiosyncrasy, namely dice overlap and mean surface distance. And these descriptors are shown, an idiosyncrasy in these descriptors is shown as a low dice overlap and high surface distance. So for example, in the visual network, we can see that this network has very low idiosyncrasy because the dice is very close to 1. However, in the ventral attention network, we can see that this network is very idiosyncratic, and because its dice score is very, very low. So when comparing both both cohorts, we can see that we found significant differences, with ASD showing to be more idiosyncratic in the dorsal and ventral attention, Samara-Moro network, and the default mode, and we found pretty much the same results with the mean surface distance, and these results also replicate across the whole cortex, as you can see here. So we also wanted to see if there is an age effect on idiosyncrasy, and as you can see from these graphs, we can see that idiosyncrasy clearly increases with age, and we found that this relationship is significant in most networks, and overall in the cortex we also find that this relationship is significant, as you can see here, that as age increases, the dice decreases, the overlap between the networks decreases, and the surface to the reference network increases too. However, when comparing both groups, we did not find differences in slope between ASD and neurotypicals. So to move on here, we move on from the network level analysis to the vertex level analysis. Here we propose two descriptors, the surface distance, in this case that we mentioned before, but in this case it's at the vertex level, and the diffusion distance, which is the Euclidean distance in the embedding space. So if we take a look at the results of these two descriptors, we can see that they do capture differences and they show that idiosyncrasy is spatially varying across the cortex. So for example, in the frontal lobe, ASD shows to have higher idiosyncrasy, whereas in the temporal lobe shows low idiosyncrasy as compared to the typical development. The same occurs with the diffusion distance descriptor here, where we still capture some differences in the temporal lobe, but here this descriptor is more sensitive in capturing differences with in the default mode, for example, here, that shows that ASD is more idiosyncratic as compared to the typically development. So because these features have shown a good sensitivity in capturing these differences, we also wanted to assess their discriminative power, and we wanted to use them to classify ASD from typically development. So in this setting, we used a support vector machine with a radial basis function kernel and a lead-one-out cross validation. So from the results and the table below, we can see that when using each feature separately, they have pretty much the same performance, but when used all together, they provide state-of-the-art performance in the classification of autism, for example, 73% in accuracy and around 70% in sensitivity. The last thing I wanted to do is to see if these scores that we proposed, in this case, dice and mean surface distance, they correlate with clinical scores. And in this case, we're using ADOS, the ADOS score. So from the graphs, we can see that ADOS is negatively correlated with dice and positively correlated with mean surface distance, or in other words, idiosyncrasy is positively correlated with ADOS. We found among the tests we have carried out, we found significant correlations, for example, with the dorsal and ventral attention networks, as you can see here. For example, on the left, the dorsal attention network is correlated with the total ADOS and communication ADOS, and the ventral attention with social and repetitive behavior scores. So to conclude, in this talk, we have presented several descriptors of idiosyncrasy based on macroscopic gradient analysis, namely diffusion distance, which is the distance between the gradients, surface distance to capture the extent of spatial shifting, and network overlap in terms of dice score. Overall, these descriptors have shown that ASD has higher idiosyncrasy as compared to the neurotypicals, and this idiosyncrasy is mainly confined to the following networks: the dorsal and ventral attention, the default mode, and Samara-Moro network, as you can see in the plots below. So finally, I just wanted to thank my colleagues at the MICA lab and also our funders. Thank you so much.